one do you think? Does anyone have a burning question right now?
Well, why
Neanderthal
don't you
DNA.
yammer a bit, and then I think we probably will have burning questions.
Okay. Wait, Jake, did you just say you had a burning question?
Yeah, that newest thing that came out about Neanderthal DNA.
Oh, okay. That's actually, there's actually not much to that study. It's although it's kind of brilliant. So that's actually been authored by a guy named Svante Pabo, who is the guy who pioneered sequencing Neanderthal DNA. That you can take fossilized remains out and still piece together human DNA out of it. So there's a project out there collecting, I believe it's like 4,000 patients at this point, where we're sequencing all the DNA from, well, not all the DNA, but a genomic profile of all these COVID-19 patients, stratified according to how severe their diseases are. And this is exactly what we want to know. Is there a genetic link between everyone who gets sick, pretty much? And it seems to be statistically a region of chromosome 3 has what's called a haplotype. What is a haplotype? A haplotype is a pattern of genetic variation that's inherited together. So you can sort of think of a haplotype as let's see, what's something that's often inherited together. Like say sickle cell anemia and Black people. Like being African is a higher probability of having sickle cell anemia. That's because the genes are connected together and they're inherited together. Although that's a pretty big characterization. That's like a really because we're talking like being African American, so it's like a whole host of properties. A haplotype tends to be a much finer resolution, so it's more modular. So think of it like I'm looking at your Jeep right now. So let's say if you're looking at cars, if this car was using the same basic design, so let's say your car uses the same basic window design as that of between a Toyota and a Ford, for example, then all the parts that connect to the window will look the same.
All the way down to the shape of the window.
All the way from the shape of the window.
Okay.
And that determines why, we would know that at least for this component, it was inherited down that line.
Okay.
And so then you can go back and then he's comparing like, okay, we know this about modern humans. We know this region of chromosome 4. Where did this region come from? And we have at least 2 major ancestral regions that we have good genetic data on. And one of them is Neanderthals and the other are the Denisovans. And so this is what's called a genome-wide association study, where we're looking at a genetic effect. We can tie it to a part of the genome, and we can say, well, where did this genome come from, or this genetic effect come from? And it seems to be inherited primarily from Neanderthals. And so the implication here is that your disease symptoms are more severe if you have this particular haplotype in your genome.
And I'm more familiar with the patterns in Neanderthal. You said Denisovan as well, they looked at?
Yeah, well, no, the Denisovans don't have this set of haplotype.
But what is the prevalence of genes in modern humans from that lineage?
So the modern humans have sort of a hodgepodge of some parts of our genomes are inherited from Neanderthals, and some from Denisovans, and the prevalence of various components varies depending on which geologic regions of the world. So it's not that, you know, they're just showing up. It's already been inherited.
Yeah, I was just wondering if you know off the top of your head, because I know the percentages of Neanderthal.
I'll have to look up that one, and I don't have it off the top of my head. Specifically for this haplotype that's from Neanderthals, it seems to be most highly present in South Asians. I think at 20% of South Asians. This is based on what's called the 1,000 Genome Project, where we just surveyed around 1,000 genomes around the world and tried to assign just sort of like a random snapshot of how the different parts of the genomes got inherited around the world. And I think based on their data, there's a 20% probability of it being in South Asians. With people in Bangladesh, up to 60% are inherit that. Somewhat lower in Europe and Native Americans, and probably the lowest in East Asians. Which is sort of an interesting idea that if you look at the death rates around the world, the Asian countries don't go anywhere near the top 12 or top 15 deaths per million around the world. Like, it seems like the East Asian countries are doing well. Part of it is probably because of just like how the behavioral surveillance of the virus has been pretty good. But part of it could also be because of the susceptibility is just not as prevalent in that population.
Okay.
Interesting.
It's kind of intriguing.
Okay, but this does not tell us exactly why this is the case, nor does it prove that it's causative. It's correlative, and there's a lot of data that goes, it's well correlated, but we have no proof that this is what causes it.
So it
We have
could
to
just be
take that with an
coincidental.
important grain of salt on that one.
Yeah.
It was kind of, publication was kind of rushed through. And mainly the big selling point there is so that people can say, Neanderthals are, you know, but really in the grand scheme of things, it's the Neanderthal part is cool. Doesn't really help us that much. It gives us sort of like a good clue of what could be causing a part of the genome that could be causing people to be more susceptible or to get sick worse. Because that's one of the things that's like really annoying about this disease, is that some people don't even have any symptoms, and then some people are dead in a week, right? And we don't really know why. Okay, so that sort of explains that study. It's actually a really short paper. Most of it was just like, okay, we did, there's, because all the data is already on databases. You could repeat it anytime. It's I make it sound like that's really easy. No, it's a lot of hard work. And that's just the coolest part, is that, you know, just like saying that, yeah, this is clearly a Neanderthal haplotype, and it's clearly unevenly distributed in the world. And that's neat. And it's correlated with disease severity of COVID-19. But it's correlated. Always careful.
Are there are other coronaviruses out there, obviously.
Oh yeah.
Is it possible that, and you know, SARS has come through, is it possible that like maybe those Neanderthal haplotypes were wiped out of parts of the world by previous, or just kind of?
That's a possibility. We have no proof of that yet. There are, we have 3 or 4 seasonally circulating coronaviruses through the human population. Those are your common cold coronaviruses. You get sick, you're momentarily immune, the immunity fades, you get sick again, blah, 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 so on and so forth. coronaviruses are a really wide range of viruses, and they circulate through all different. Okay, so let me wind back. This is where I kind of wanted to start with. What is a virus?
Oh, are we
Is
supposed to answer?
Yeah, well, I wanted to see what people's idea of what a virus is.
It's a packet of DNA that is not self-replicating. No, it isn't always DNA. It's a packet of genetic material that is contained. I think of it sort of as in suspended animation until it manages to latch on to the cell from another organism, which will then replicate the virus instead of its own.
What do the rest of you guys think?
I'll go last. Let's let Tracy go next.
Oh, so I kind of go along with what Catherine said. I mean, basically, they have protein codes. So that's the only thing I'd add to it.
I would go with an incomplete living organism. Because
I'm not even gonna argue that they're alive.
Well, but that's what I'm saying. I'm saying they're incomplete because they need something else to replicate. Without the help of something else, they are in suspended animation, like you said. They're basically a non-entity until they are.
All right, I'll differ with you in what an organism is. And what
Okay,
that's fair.
So here's the part that I wanted to point out that all this discussion is that everyone talks about viruses that's if they're separate from us.
Yeah.
But they aren't. What so we humans have this we're such social creatures that a key defining factor in any discussion we have is something part of us or separate from us.
Okay.
Right. So we talk about this a lot. We talk about like, are they part of us? Are they part of this group? And even when we talk about biology, the first thing that we talk about is classification. We put things into containers, into boxes, and viruses are containers. The reality is that viruses aren't really separate biological organisms. And so, but we call them viruses because it's sort of like a catch-all phrase. A bacterial virus has almost nothing to do with a polio virus or a coronavirus, but we call them all viruses because they need to fit in a box. It's like our junk drawer of these things. And what it is life is messy. They don't fit in boxes, and genetic material does. Wow, that was impressive. We just did the whole. This is like the modern seance where everyone does holograms and stuff like that. Anyway, so viruses are anything that is not a biological, like a regular biological process, communicating between individuals. So the idea of the selfish gene, where individual genes, where evolution can act on single molecules, not on just whole organisms, but on individual molecules, is a key idea here. Viruses are a product of evolution acting on individual molecules, where a genetic molecule says, like under genetic pressure goes, can it replicate without depending on the rest of the cell? It'll do it.
Okay.
So you give it that niche and it'll do it. So Catherine, you brought up a really important point. It's not always DNA.
Yeah, it's RNA too.
And in fact, for a lot of the vertebrate So here's another thing. Like when we talk about viruses, we talk about viruses that are important for human illness. So things that infect vertebrates, things that infect animals, usually, mostly. And the majority of these sort of pathogenic viruses are RNA-based viruses, not DNA-based viruses. But there are a lot of important DNA-based viruses. And most of biology, when we talk about classification like animals and plants, there is an evolutionary link between all of them. So if I'm going to talk about the different animals, it's because they are related to each other in evolution. Right? This does not tend to be true when we just lump viruses together.
We lump
because
them
the viruses, sorry.
We lump them by what they do to us?
Do we lump them based on the organism in which they are native to?
Oh, interesting. Okay.
And what they're native to is only a matter of, and those organisms are connected by evolution.
So here's a
But
question
the
for you.
nice thing about viruses is that they can just like cross those lines. They just jump across. So bird viruses can jump across species and start infecting mice, or you know, from bats are our favorite little thing. So coronaviruses circulate among a whole bunch of vertebrates, bats, birds, rodents, definitely. I think I just was reading about the fact that I think we've underestimated the potential for rodents to be a virus reservoir. Like all of these different viruses can circulate in that area. And when you have a reservoir, when you have just like giant pool of information and evolution acting on it, that you can hit the, you know, that magic 1,000 monkeys. Is that 1,000 monkeys? Where if you let them give infinite time, they'll eventually type out Macbeth, is it?
Right.
Or
Shakespeare.
Or you know, one of the Shakespeares. So like, I can just hear the monkey typing, like, "This is the Tempest. God damn it! You know, go back.
Start over."
Macbeth. Okay. Anyway, you have infinite time. Go do it. So that's generally the problem with reservoirs, with genetic reservoirs, is, and that's why we're monitoring the bats and all the situations, because eventually these reservoirs of viruses, if they have the potential, if they're close enough to humans with enough contact, and that they can make the jump and propagate in humans, and then it's a crapshoot whether it can cause a disease or not.
So just for curiosity's sake, you kind of hit something when I was thinking. So when we say viruses as a whole, if we're talking about DNA versus RNA-based viruses, that almost sounds like it'd be as different of a breach as the difference between eukaryotic and prokaryotic.
Oh yes, but fortunately, here's sort of like the difference. This is the thing that's like will blow most people's minds. Prokaryotes and eukaryotes are genetically related.
Right.
Okay, this has no evolutionary relationship.
So does that mean separate origins, entirely separate origins?
It's not just entirely separate origins. The concept is polyphyletic, and they're originating all the time. Yeah. Viruses are the messy side of biology. Like if you look at, we like to think of it like, oh, look at, the plant is a plant, well, not even a plant, it's a plant. Like this is a fir, this is a kelp, this is a rabbit. But what's going on the whole time on a molecular scale is that molecules are taking every opportunity they can to find the next opportunity to just make more copies.
So not only is it different origins, but what I'm hearing you say is we've had a lot more recent origins for some of these viruses than 4 billion years ago, or 3 billion years ago. 3.8.
You can say viruses have always been with us.
Yes, but have we had totally new viruses sort of pop up out of the primordial ooze after?
You mean like just de novo built? We can build that from in the lab anytime
Well,
we
we
want.
can, but has it happened in nature, or can we even tell?
We can't ever prove that.
So what you're saying is that viruses could almost be they could almost be spontaneous generation as far as the idea of they just.
Yeah, except they're not really alive.
Right, kind of like with, subatomic particles, just, appearing and disappearing.
Like, as long as life exists, and as long as you're able to do natural selection on single molecule scale, something like a virus is going to arise. Tracy, to address your question, there's a variety of viruses called viroids that have no protein coat. It's literally just naked RNA, and you can put it on the organism, and it'll infect it.
How do they survive? I mean, how do they defend themselves against the outside world?
It doesn't need to.
It's
Really?
in the body.
I thought RNA is not stable outside of basically,
You'd think that, but I think that comes from a laboratory bias.
Yeah, I couldn't keep it stable.
As it turns out, we are able to get SARS-CoV-2 RNA out of sewage and forecast when the next outbreak is going to happen.
Right.
Just based on increase, and that's RNA in poop. Like, how much messier you can get, it's obviously stable enough to survive that.
Right, but I was assuming those were just fragments that you were looking at in there.
Fragments is all you need. It's all fragments at the end of the day.
Well, okay, fine. But I mean, you need enough basically of the package so that you can actually, eventually when it reaches a cell, express itself again, right?
In one form. That's a beautiful and messy thing about evolution.
Mm.
If you mash enough things together with a large enough population, eventually something works.
Okay.
So something like a viroid folds itself into these various conformations that seem to protect it.
Oh.
And keep it from being degraded so easily.
What degrades it? Is it oxygen or?
No, we have specific enzymes, RNAs, and it's just that. So it's probably an interesting point. So we love to think of DNA as the big kahuna of molecular. Like this is DNA, like this is the DNA. Most of the world is actually run by RNA. We actually think that the origin of life probably started with RNA.
Yeah.
RNA can encode information. RNA can do almost all the enzymatic things you need to do. Mm-hmm. All the protein is made in your cell by ribosomes. And people used to think that ribosomes are big protein complexes. Wrong. All the proteins do is hold the RNA together, and the RNA does the actual work. So RNA can catalyze enzymatic reactions. RNA can encode the information. And does everyone know about the central dogma of molecular biology?
Mm-hmm. No.
Okay. Central dogma of molecular biology says DNA makes RNA, RNA makes protein.
Oh, yeah.
Okay.
And protein does everything.
Right.
Okay, that dogma is definitely just dogma.
Yeah.
Because it turns out that RNA can do everything. And one of the things that's really important to understand about something like coronavirus is that DNA is never involved. RNA, the coronavirus goes in, it makes it uses the cell, translates it to make an RNA-driven RNA polymerase. It can make RNA from previous RNA. Mm-hmm. DNA is never involved in the whole process.
Why is that important?
Sorry?
Why does that matter?
It matters because then when you have a cell where it's a eukaryotic cell, the nucleus is sort of the DNA is protected in a separate compartment,
Mhm.
right? So usually, like when people talk about our viruses coming in and then sort of commandeering the replication process, all this replication process is in the nucleus.
Mhm.
But this doesn't need to
This,
go into the nucleus, so it can sit in the cytoplasm of the cell and do its job there.
Mhm.
oh, at the right moment, so it's been measured from the papers I've read. Once SARS-CoV-2, this is probably true of other viruses, it can take it can take over 68% of the entire RNA output of the cell.
Mm.
So it sounds like it's easier for it to infect on a larger scale than it would be if it were just focused on the DNA.
But like if you don't need to use the DNA machinery and you carry your own machinery and you already are the genetic material, then you just go gangbusters.
Right.
Okay.
So there's no need for reverse transcriptase. You don't need to basically go into the nucleus. You don't got to do any of that stuff. So yeah.
Well, right,
So-
but I'm saying, cool. That's cool, but the net result is it's easier for it to have a bigger effect. Is that accurate?
Conceptually, yes. Although there's a lot of other barriers along the way.
Mhm.
Not thinking that at the end of the day, the virus doesn't really want to kill the host.
Right.
Because if it kills the host, then it's the end of the line for the virus too. So we're trying to find an equilibrium where there's enough of the virus that goes around and enough of the host to go around, so that they exist going forward.
Mm-hmm.
But this is an important thing. Whenever you hear people talk about coronaviruses and they talk about the DNA of the coronavirus, your alarm bell should go off right away. Like there is never DNA involved with coronaviruses.
Is that's part of the reason why it's so hard to make a vaccine for them, right? Is because they're RNA based.
No.
Oh, okay.
Oh, it's hard to make. We actually think that this is relatively easy to make a vaccine for. It's just hard to make vaccines, period.
Mhm.
Okay.
My impression was that RNA viruses, and this is like from my classes way long ago, mutate or at least are somewhat prone to mutate at a higher rate.
Good point. The reason is because RNA is a little more unstable and they tend to recombine and they do all sorts of weird things. Coronaviruses are a little weird. So RNA viruses they're viruses, they're small, so they get really I love how elegant coronaviruses are structured, and when they I can go through like the contortions it goes through to package it, but most viruses are small. We're talking 2,000 bases or something like that, because there's only so much you can put in there. A coronavirus is 30,000 bases long.
Wow.
And part of it is because, and the reason why it's called a coronavirus is because it has this protective shell that's not just RNA, and it's not just protein, but an actual membrane.
So it's almost a cell.
It's almost a cell in that sense, except once you peel this membrane out, the membrane serves a role. So the shell has these things called, you must have heard the term spike protein, because it has these spikes sticking out, and that's what gives it the term coronavirus, because if you look at it under electron microscope, it looks like the corona of the sun.
Fair enough.
The spikes are sticking out. And the spikes...
if you've never seen the corona of the sun, but...
Yeah, well, okay.
Technicality.
As long as you don't talk to an astronomy teacher, it's fine.
It looks like the corona drawn by a 3-year-old. We're good. It's...
Or or somebody who stared directly at it like a president.
Okay, there you go.
Uh,
Sorry, I interrupted you.
no, we have so many. I have so many things to say about this. uh,
So before you get on, as long as we're interrupted, I have to
no,
split.
you're
So
not
thank you
okay.
for doing this. But yeah, Catherine, I'd be very interested in the recording. Okay.
Sure. Well, Tracy, we were arguing, but I don't remember about what. Not arguing, trying to discuss it. There was a question that you were trying to figure out about the.
Yeah.
Actually, Dr. Ricky, one of the things I was wondering about was epigenetic modification of the RNA, but that's a really big topic. And
That's a great question.
yeah,
Unfortunately, we don't know very much about epigenetic inheritance of RNA.
okay,
Epigenetics is usually what we know about the molecular mechanism of epigenetics is on DNA.
right.
So it sounds like methylation.
Yeah.
We don't know enough about epigenetic modifications of RNA-based encoding yet.
Okay. All right. Just figure I'd check out with you on that. Anyway, thanks, and I'll check out the recording. So see you guys next time. Then sorry
Good to see
about
you, man.
Okay, sure.
Bye.
Tracy's leaving, but the other thing he was asking if I had seen anything about was whether the virus is in fact getting less lethal percentage-wise with time, or whether it's a statistical phenomena or medical interventions.
Oh, that's kind of ongoing. We don't have enough data for that. We have to posit that, because it's not really the virus changing, it's humans changing too.
Fair enough.
And we know that there is a lower rate of, we're becoming, people have a better chance of surviving now, because we have better medical interventions. We can detect them sooner. People are exposed to lower viral loads, and the amount of virus that you are inoculated with has an effect on how severe your symptoms are.
Is there also a more morbid possibility that the people that are most susceptible have started not being as common?
We'd like to say that, but I think the population size is still too big for that purpose. Yeah, I mean, if you let it run the long course, and we probably at the point when we've like killed off like 1 million Americans, that might be true.
Well, I suspect there's a few nursing homes in Kirkland out here where that might be true. But
are they current nursing homes or former nursing homes, because they've been killed off?
They're still in business, but what, it was something like 30% of the residents of one place, the place where before they actually quite figured out what was going on.
Yeah.
But there's a lot of other nursing homes in Washington still.
Yeah.
Anyway,
because the problem with this is, of course, like the pathology is not consistent. So some people, with no intervention, or will be just fine. Some people it's more severe. We don't know if that is inherent to the patient, or the exposure time, or how they're treated. Like, let's say somebody was a smoker who had just quit, or someone who was obese, who was like getting exercise, and so on and so forth. We know there are risk factors, but the problem with these risk factors in the medical profession is that it's a snapshot in time.
I knew that, I mean, obviously smoking has long-term effects. It sounds like your possibility that obesity likewise has long-term, I guess there'd be cardiovascular implications of it, even if you lost the weight.
Right.
That's true.
Okay.
So we don't actually think obesity itself is a direct factor for it, but obesity comes linked to all these other medical scenarios, and any one of them can aggravate your symptoms.
Well,
right, and just because
makes your body work harder too.
Yeah, but just because you've lost the weight, especially if it was recently, doesn't mean that the damage to other systems has.
It's easier to explain in like immunocompromised patients.
Right.
And that's one thing that I'm hoping to get more data on because one of the research projects I want to get at is whether the viruses diversify more in immunocompromised patients than not. Because it would not put them through a selection criteria. So you would have a wider pool of genetic variation in it. I have a little bit of evidence, hopefully, that it gets published soon, that this might be going on.
Interesting.
But
would that make it easier or harder to treat? I guess it's a little bit of luck of the draw.
For the individual patient, they're already immunocompromised. You treat them however you can treat them, because they're immunocompromised. What this does imply on a population scale is that as this virus becomes endemic to the human population, this might actually be your incubators of genetic variation going into the future.
What do you mean?
Where in evolutionary biology, an area with greater variation means that they can adapt that population can adapt to a new environment faster.
Right.
So if you have a population that is if you have humans who are immunocompromised who can incubate a greater variation of coronaviruses, if you immunize everybody against this current virus, then they would likely be capable of incubating the version that can get past this immunity.
Okay.
In fact, that's why we think bats are such a hotbed of coronaviruses, is because their immune system is a little unusual, and it might allow for the viruses to incubate greater variety.
Why do bats have weird immune systems?
Evolution. I need to go and look up the specifics of it, but there are certain genes that are lost in the bat lineage, and there are also between bats and rodents that accounts for 2/3 of all mammalian diversity.
Right.
So there's a lot of them.
Yeah, I had no idea they had different immune systems. So interesting.
That was actually known, and that's the reason why the CDC had a team in China monitoring the bats.
Right. I knew bats were
even well before this.
Interesting.
And if Peter Daszak's work gets picked up again, hopefully we'll pick up the 4,000 samples he has in the freezer, because he published 400 additional strains that might go pandemic. Just 400 more.
So these are strains he found in the bats that he says, oh yeah, by the way, we should watch these too. Interesting.
Yeah. He had to publish them because they yanked his funding earlier this year before he could finish the whole thing.
Yeah, always brilliant.
So brilliant. I'm petitioning that so one thing that this whole super spreader White House event would happen, and I hope we could write our different Congress people and everything, is can we ask that all the people who've tested positive in this agree to release the genetic sequence of their viruses to the scientific community?
I like it. Yes.
Not asking them to release their genetic information.
No.
The genetic information of the virus that's was isolated from them, because for once we would have a key event, one location where we know how we can trace the transmission rates and how it changed, and just release that if it's the same one. It would be valuable for epidemiology, and it would make this sort of sad event useful.
Yeah, it's just I'd be interested in just even seeing if it is the same. It seems to be, but just having that information.
We would have, so this is one of the things where the patients have to consent.
Yeah.
Right. So this is where, you know, we run into this problem all the time. Like we before we can test, so the for our testing group, we require a protocol, and we consent, the patients have to give consent that their information can be used for genetic analysis. Like I would like it. I mean, this would be a very valuable supplement. This would be like, you know, and just release it for public access.
Right.
Yeah. These are the samples. We'll sequence them, and we'll, you know, it can be done in under a week. But, you know, Lord knows what. Anyway. I think I hear that. Okay. Well, oh, I think I dropped off on this one. So, viruses. The coronavirus is 30,000 letters long. It's unusually big. Right? What makes it capable of being this big is that it carries a specific enzyme that does proofreading.
Oh, okay.
Which is to say that as it's replicating, it goes back and checks to make sure that it hasn't made an error.
Right.
This is a feature that's missing in most viruses, which is why they have to stay short. Because once it gets a little bit long, then the errors will rack up, they'll split apart, and you get two different viruses. But there's a proofreading enzyme, so you get to be really big. But the other thing that's also interesting about having a proofreading enzyme is that the fidelity from generation to generation is pretty good. And this is an important factor. So as far as viruses are concerned, coronaviruses don't change as much as other viruses.
Very interesting.
So we actually do think that we have good hopes that we can generate a vaccine for this. Okay. What people kind of forget is that vaccines are difficult to make in general.
Right.
We're trying to produce a vaccine in under 2 years, which is twice as fast as the fastest vaccine ever developed.
What's the fastest previously? What's the fastest?
I forget which one.
4 years.
Mumps for 4 years.
Yeah.
Polio took 50.
Well, there's some history and politics in there, but yeah.
Oh yes. Oh yes. So we're talking about vaccines.
Sure.
Politics never interferes with science. What are you talking about?
Oh. So vaccines. This is sort of an interesting one. I think important for as you teach your students about this. So when people talk about vaccines, they assume that vaccine. What do vaccines do?
They help train your immune system to recognize and deal with a specific intruder.
Right. So, so it deals with it. It primes the immune system, but people usually assume that the vaccine is what's called a sterilizing immunity.
Right.
What does that? That means the infecting agent comes in, it stops it, and that's where you get your immunity because it stops it at the door. Make sense so far?
That's not the case in any situation, is it?
Well, in medicine, the priority isn't stopping the infection. The priority is in stopping the disease.
Yes.
So vaccines, our priority is actually immunizing against the disease, against the symptoms, not necessarily the infection. So just because we can have vaccines that confer immunity against the disease, but not against infection.
Can you give an example of something like that?
Yes. So polio is a great example of that. So there are two types of polio vaccines. There's one that's injected, and there's the oral vaccine. So do you know where polio is, how polio actually works, and why you get? So I'm sure a lot of kids don't know how horrible polio was. I hope that someday when you have, like, show them pictures of iron lungs, and in fact, we considered breaking out an iron lung here for the patient of COVID-19 here.
Oh, wow.
But so polio is actually a gastrointestinal virus. It infects the lining of the gut. And once in a while, it escapes that area and it hits the central nervous system. And that's when you get paralysis.
Yeah, I actually knew someone growing up who had residual paralysis. So I doubt there are very many people under 40 in this country who could say the same thing.
Oh, God. One of my wrestlers' dad was actually infected during the live vaccine trials in the 60s. And his whole leg is... Yeah.
But again...
Yeah, but sort of like... But it's kind of like what's going on right now with COVID-19 is that there's a range of symptoms. For some people, if you're lucky, you get some diarrhea and you're done. And some other people, you lost the ability to breathe.
What percentage of the people are relatively mild for polio? Do you know?
I will have to look that up. But
I'm sorry, we're getting off on a tangent. It's just fascinating.
I think it's actually the majority. So it's like that luck of the draw thing. Okay, but even if like 10% of people or 5% of people, I was social distance from everybody, and so there are the two vaccines. If you get an intramuscular injection of the anti-polio vaccine, you would have circulating antibodies against polio. But what that does is it doesn't prevent you from being infected in the gut, which is where the polio virus actually infects you. You can, in fact, produce more virus. You're infected. You won't have symptoms, but you can shed virus.
Right. So you can be a carrier.
You can be an infectious carrier, and that's what it does. That's perfectly fine vaccine, but it's good if everyone gets the vaccine. Because what happens is that if you only vaccinate a small population or not enough of the population, you can actually increase the infection rate.
Yeah, because you have all these people shedding virus out.
So right. In the United States, with people saying that, about half the population says they're not going to take a coronavirus vaccine if it gets produced, half the people getting it would make it.
Right.
Wow.
Interesting.
So this is an important case note that the oral vaccine causes an immunity in your gut, and so it prevents the virus from infecting you there.
This is always something I wondered about polio, the oral vaccine. Do oral vaccines only work for gastrointestinal diseases? Or...
Well, you would do an oral vaccine if it's something that's specific to that tissue. If that is if the virus is specific to going through that tissue, because you want to block it there.
Right.
But
so an oral vaccine won't work if it's lung disease, say, for example.
Yeah. So we would actually want to do something like an inhaling vaccine or something like that. But again, there are issues with this. An oral vaccine has to survive the gut and has to go through all the way, and in order to ensure that you get that rate of immunity, it might not be there. So the intramuscular one is a little more assured, but it doesn't stop you from being infected or shedding virus.
So is that the same with measles and stuff like that?
No, I think measles, I need to check with measles, because I think measles can actually infect multiple tissue types. So these are the polio ones are just a good example because it has a very specific tissue type that it goes through.
Right.
It's the same thing with SARS-CoV-2 for COVID-19. The primary mode of infection is through the nose, but we have evidence that it can infect through the gut.
Okay, so that
So-
would be one reason why there's such a range of symptoms.
Right.
And
that's why we can pick it up in sewage, is because people are shedding it out of the gut.
I had wanted actually to flag this article to you guys after your discussion with the face shields, Ricky, is the paper that came out suggesting people who wear glasses have a lower infection rate.
Yes, it actually was that came from China.
Yeah.
Which I thought, it's again another correlation study. It's a retrospective correlation study, but the numbers are pretty good.
Yeah, it seems solid. I was dubious when I saw the headlines, and then from what I could tell, I mean, correlation versus causation is a separate issue, but statistically, it didn't seem as fly by night as some of those.
No, it kind of makes sense. The model hangs on itself, which is why people aren't too nuts about it. But on the other hand, it's still correlation.
Yeah.
Right? We need to sort of like take that with a grain of salt and but the definitive experiment will involve actually infecting people, and you kind of don't want to do that. Yeah.
Well, it depends. Yeah, never mind. I'm not going to say anything. This is being recorded. Better not say anything.
Let your evil scientist out. Here we go.
You just get little glasses for your lab rats and have half of them with goggles.
Oh, lab rats. This is interesting because lab rats can't be infected by SARS-CoV-2, right? Because it's
not yet.
But what's his name? Jeff Barrick has developed mice that can be infected by SARS-CoV-2 and had to develop a strain of SARS-CoV-2 that gives mice COVID.
Yeah, I guess so.
So that's fairly recent, like within the last week.
How about I just have lab bats and I put little goggles on my bats?
They don't really need to see.
That's not the point.
Bats have just as good eyes as you do.
And there is a group that's actually doing lab bats.
Okay. Oh boy.
The problem with lab bats is they need to fly.
Right.
And they're studying a highly infectious virus that causes the humans. So they need to be in what's called a BSL-3, a biosafety level 3 facility. And space is precious for biosafety level 3 labs. So they're actually trying to petition to build a bat cage in a biosafety level 3.
Oh my goodness.
Can you imagine the cost involved in something like this?
Bat cage or bat cave?
It's a bat.
I'm not sorry. Ricky's having problems.
He's
Moving on.
Where's the pumice? See, we just need to issue to every student a little box that they put next to their computer that they have to preload, and you plug it in. You can hit a button and then just poof.
Right. You want them to honorably do that.
I'm gonna have to bail. So,
okay. Yeah, we're
yeah. Hey, last question for Ricky then. Have you been able to figure this out? Because I have not. What the prevalence of long-term complications is? People are making a big deal about correlations with ME. And I'm not convinced yet because they say long haul is 3 weeks, and if it's anything under 2 years, I'm not going to give them.
The disease is less than 8 months old.
Yeah, exactly. So even if we know for 6 months, I mean, there's lots of.
We're still the definitions are still kind of evolving, medically speaking, because the disease is so young.
Yeah.
And you're asking for long term. So I love people kind of tell me like, "Well, the long term results are like we don't We didn't. This
Yeah.
didn't even have a name until February.
Yeah.
Like, really, this is we're learning. We're building a plane as we're flying it. Like, this is what we got. So, is there a correlation? The prevalence we don't know yet. It might be possible that people who have recovered from it won't show sort of like the secondary effects until
True.
like 2 years from now or 3 years
Well,
from now.
what my question is so there's lots of attention to folks who are still feeling the effects
Yep.
a few weeks or a few months down the line, but is it any different from any other disease? Because almost anything out there, including some are worse than the others, but almost anything out there, you can have a reaction with your own immune system that will.
I think that's really weird about this is that we have not. I don't think anything has described this wide range of symptoms to one causative pathogen ever.
Okay. I am going to get
in this short.
ditching before.
Okay, yeah, we're just finishing up.
No,
So.
go ahead as long as you want. I mean, it was great hearing, and I want to hear more, but
I'll post the video.
We can do brain eating amoebas at some time.
Nice. All right, cool. Good seeing you guys. I will talk to you soon. All right.
Take care. Bye. There's a lot of I don't know yet there. We'll learn more as it comes along, because it's easy to go into all these areas, but we're still learning it. We don't
Yeah,
know.
well, that's the potential for long-term complications is something, as you say, we don't know yet, but is one of the more worrisome things potentially.
I mean, it's really scary.
Yeah.
If you are that patient. So Ian Lipkin, who is also one of the world's leading virologists, is one of the long haulers.
Okay.
And
Wow.
and he describes it as, he still loses his breath going a flight of stairs.
Mm-hmm.
It's like he suddenly lost more than half his lung capacity,
Mm-hmm.
and it's been months.
Right.
Like, it could be that this is what it is for the rest of his life.
Yeah, well, I gather the folks in Italy who have been released from the hospital have been studied, and it's a scary number with reduced lung capacity. But there's also just sort of immunological responses.
The things that scare me are things like the blood clotting, the chances of stroke. That affects your probability of stroke going in the future.
Yeah, good point.
That's, like, suddenly your whole life is a ticking time bomb.
Yeah, yeah.
Like, that's not a great place to be.
Yeah, well, I mean, it's heartbreaking how little attention, the 200,000 plus people who have died in this country have gotten. And beyond that, there's this whole other, I don't even know what you pile of worry.
The 200,000 are just the official count. I believe the number is probably at least double that.
Yes. And out of curiosity, I was paying very close attention to the unexplained, what do they call it? Unexplained death, essentially, or unexpected increase. So people in New York City. Excess death, yeah. So are you including people who died from other causes as well in that half, or just the official count of the coronavirus victims?
No, 200,000 is just an official count.
Well,
it's the official.
Right.
But I think if you look at excess deaths, that just looks at death, period.
Right.
And what you do is you do a statistical projection and say the prior year at the same period of time and the current year, this is how much excess death there is. And it's all over the world. And it's roughly the same proportion all over the world. And the only thing that's happening all over the world right now is the pandemic.
Right.
So you deduce that this must be all of it. And it's definitely higher than the 200,000.
Right. And
everyone who's died who didn't make it to the hospital in time.
Right.
Or died before they could get tested, so on and so forth.
So yeah, so you're when you're saying twice as much, you're talking about the excess death.
Right.
Yeah. No, I knew a lot of people in New Jersey and New York. They're talking about one of the difficult folks things for folks who are isolated but were not sick was hearing the sirens constantly from them taking the people to the hospital, or a lot of cases not making it in time, but they'd still have to go with the sirens. Anyways, that's a rather grim place to end, but.
Well, Houston dodged a bullet in that regard from July and August. It was deathly scary when
Right.
your positivity rates are 20%.
Any idea how you dodged that bullet?
Partly it's like implementing the mask mandate in time,
Okay.
and the hospitals were just on call. There were a lot of heroic efforts that will never get acknowledged.
Yeah.
There's a New York Times article that looked into how they managed to get it done. It was actually pretty.
Okay.
It didn't get, aside from just like one embedded reporter who actually came from covering the New York situation and came down to Houston, and where our staff were all like preparing to be inundated by the same problems.
Right.
In my opinion, the only hiccup there is when our idiot of a governor decided to intervene.
Yeah. Well, I wonder in part for the New York, but like you say, they didn't have a mask mandate. They didn't. I think my impression is the evidence was there for the masks, but that was a failure of politics and government and communications. But in addition to that, my impression with New York is it was much higher level of prevalence in the population, so it had spread much farther before they first recognized it.
Yes.
Do you know if that's true?
We have evidence in New York of multiple introductions into New York. So places like Houston, Seattle had one. I think you can trace it to one index case, I think, or
Possibly
two. Possibly two. And it only spread because it's the first one. We didn't know anything about it. I think the estimate for New York is at least 40.
Uh-huh. And my impression is that they were probably introduced at about the same time or shortly after Seattle.
Well, it's a traffic from, so you can, based on the genetic information, Seattle is from China and
Right.
New York is Europe.
Right.
So it's probably the traffic coming in from Europe as it was spreading through Europe. And yeah, so they were roughly around the same time, but that's also like when it was blowing up in Europe.
Right. But in part because I think it tracked up to the north first, and it actually hit Kirkland nursing homes. And because it was that population early on and really lethal, Washington jumped into gear before New York did in terms of testing.
Also, what's the name of the university in Washington? The Fred Hutch.
Yes.
Had a team monitoring it, and because it was only at most two index cases, it had that sort of like logarithmic slow burn up.
Right.
And by the time they reported on it and figured out based on the molecular clock, and it actually was corresponding with the science, it's been circulating for 6 weeks.
Right.
In Washington. New York was just kind of like la-di-da, we got lots of people coming through, and people were using subways, and and just blew up.
Yeah.
So, but they did what they could do.